Friends, today we'll study about Mycobacterium tuberculosis. Under this, we'll study about the introduction, then classification, tuberculosis, morphology, culture, resistance, biochemical reactions, antigenic structure, mycobacteriophage, phages, and pathogenesis. Next, we'll study about immunity and hypersensitivity, cause phenomenon, tuberculin test, laboratory diagnosis, sensitivity testing, treatment, prophylaxis. Now let's study about the introduction. Mycobacteria are slender bacilli that sometimes show branching filamentous forms resembling fungal mycelium. They are difficult to stain, but once stained, resistant decolorization with dilute mineral acids and are therefore called acid-fast bacilli or AFB. These organisms are aerobic, non-motile, non-capsulated, and non-sporing. Growth is generally slow. The genus includes obligate parasites, opportunistic pathogens, and saprophytic mycobacteria. Now let's study about the Hansen. Who is Hansen? Hansen is the scientist in 1868 discovered the first member of the genus, that is Leprabacillus. Robert Koch in 1882 isolated the mammalian tubercle bacillus, and John in 1895 described M. paratuberculosis, causing chronic enteritis in cattle. Subsequently, other pathogenic tubercle bacilli such as the murine type, that is M. microti from voles, the avian type M. avium from birds, and cold-blooded type M. piscium, M. marinum were isolated. The saprophytic mycobacteria were isolated from a number of sources. Example: butter, M. butyricum, grass, that is M. clayi, dung, that is M. stercoris, smegma, that is M. smegmatis. A group of mycobacteria distinct from human or bovine tubercle bacilli, isolated occasionally from human sources, are grouped together under the term atypical mycobacteria. Anonymous or unclassified mycobacteria. Most of these bacteria occur in soil, water, and other sources. They are mostly opportunistic. Four closely related species are responsible for mammalian tuberculosis: M. tuberculosis, human tubercle bacillus, M. bovis, that is bovine tubercle bacillus, M. microti, vole tubercle bacillus, and M. africanum. Intermediate form between M. tuberculosis and M. bovis. Now let's study about the Mycobacterium tuberculosis complex. The M tuberculosis complex includes M. tuberculosis, M. bovis, M. microti, and M. africanum. These four species are so closely related to each other by antigenic analysis and DNA hybridization that they are regarded by many variants of a single species. However, they can be distinguished by certain characters. Now, M. tuberculosis and M. bovis are typical tubercle bacilli and cause human lesions such as pulmonary tuberculosis. Now, let's study about Mycobacterium tuberculosis. The species contains two major types: classical and South Indian type of M. tuberculosis. Classical type is virulent to guinea pig, but South Indian type is attenuated in this animal. South Indian type is prevalent in South India and in persons of Asian ethnic origin living in other countries. Now let's study about the morphology. M. tuberculosis is a slender, slight, or slightly curved bacillus with rounded ends occurring singly, in pairs, or in small clumps. It measures 1 to 4 micrometer into 0.2 to 0.8 micrometer. Average 3 micrometer into 0.3 micrometer in size. These bacilli are acid fast, non-sporing, non-capsulated, and non-motile. Gel Nelson staining is useful to study the morphology of these organisms. With this stain, tubercle bacilli are seen bright red, that means acid fast, while the tissue cells and other organisms are stained blue. Tubercle bacilli may also be stained with the fluorescent dye, that is auramin O or rhodamin, and appear yellow luminous bacilli under the fluorescent microscope. Beaded or bad forms are frequently seen in M. tuberculosis. They are gram positive but are difficult to stain with gram stain due to the failure of the dye to penetrate the cell wall. The M. bovis appears stouter, and shorter with uniform staining. Now let's study about the culture. M. tuberculosis is an obligate aerobe, whereas M. bovis is microaerophilic on primary isolation, becoming aerobic on subculture. The bacilli grow slowly, generation time 14 to 15 hours, and colonies appear only in about 2 weeks, and sometimes it may take up to 6 to 8 weeks. Optimum temperature for growth is 37 degrees centigrade, that ranges between 30 to 40 degrees centigrade. Optimum pH is 6.4 to 7.0. Tubercle bacilli can grow on a wide range of enriched culture media, but Lowenstein Jensen medium is most commonly used. This medium consists of beaten eggs, asparagine, mineral salts, malachite green, and glycerol or sodium pyruvate. It is solidified by heating. In speciation, it is one of the media which is solid without incorporation of agar. In this medium, egg acts as a solidifying agent. Malachite green inhibits the growth of organisms other than mycobacteria and provides a color to the medium. The addition, the addition of glycerol and improves the addition of glycerol improves the growth of human type of M. tuberculosis, while it is without any effect or even inhibitory to M. bovis. Sodium pyruvate improves the growth of both M. tuberculosis and M. bovis. Colonies of M. tuberculosis are dry, rough, buff colored, raised with a wrinkled surface. They are tenacious and not easily emulsified. In contrast, the colonies of M. bovis are flat, smooth, moist, and white, breaking up easily when tossed. M. tuberculosis has a luxuriant growth in culture as compared to sparsely grown M. bovis. In liquid media, the bacilli grow as surface pellicle due to hydrophobic properties of their cell wall. Diffuse uniform diffuse